Hey guys, welcome to CakeWalk Cambridge. I'm Aisha and today we're starting with chapter 13, Excretion. It's raining today and it's really dark, so that's why the lighting sucks. But yeah, my face doesn't really matter like the content does, so don't judge me right now. Let's get started. Okay, so in the first three things you're supposed to be able to state in the chapter is that urea is formed in the liver from excess amino acids. This happens in a process called deamination. But what you need to be able to state is that the liver is where urea is formed, and urea is formed from excess amino acids. Excess means in surplus, in extra supply. So when there are excess amino acids, the liver deaminates them to form urea. Okay, we'll learn that whole process. Carbon dioxide is excreted through the lungs, and kidneys excrete urea and excess water and salts. So very often you have a table in your MCQs which gives you like a chart kind of um, structure where they'll give you carbon dioxide, so like carbon dioxide, urea, excess water and salts, and then lungs, kidney, liver, etc. And you're supposed to tick where it is excreted. So just remember, kidneys excrete urea, excess water and salts. The lungs also excrete excess water via water vapors, in the form of water vapor via respiration, as we learned in chapter 11 and 12 that air breathed out has saturated water vapor. Water vapor is merely the gaseous form of water, so the lungs also excrete water and carbon dioxide as well. You just have to be able to state these three things. They're quite easy to learn and understand, okay? So those are your first three requirements of the syllabus. Now this is the structure of your excretory system. Okay, so you have the aorta, which is basically the largest artery in your body. It is the artery from the heart that we have studied before as well. So the aorta, it carries oxygenated blood, transports oxygenated blood. So the aorta branches into the renal artery. So as you can see, oxygenated blood is denoted by red color, okay? And deoxygenated blood is denoted by blue. So the aorta branches into the renal vein, which then enters the kidney. Okay. You have the vena cava here, which is the vein that takes the oxygenated blood from all over the body to the heart, to the right side of the heart. So what happens is it basically has the highest concentration of carbon dioxide. So what happens is this also branches into both kidneys. Okay. Then you have two kidneys which are bean shaped. Okay, that's how you describe the shape or structure of a kidney. You have two long ureters which are like two pipes from the kidney that join to the bladder. Okay, the bladder is like an organ that fills up with with urine, and once the body is ready to excrete the urine, there's a sphincter right over here, around here, which kind of relaxes. Otherwise, it's contracted to control the movement of urine through your body, through your urethra. So it relaxes, and the urethra opens, and your urine flows out. Okay, so that is it. Um, the renal artery has the highest concentration of urea, and the renal vein has the highest concentration of carbon dioxide. You should be able to state both of these. They've come in, I think, a 2015 paper earlier, which I got as my mock, and a lot of kids got this wrong, but that's what you should be able to know. Okay, I think this was pretty easy to understand. Okay, important, you should also know the drawing of the kidney and the ureter. Very often you're asked to draw it, okay? So you should be able to identify it. Also, if you want to, so your Cambridge papers don't give colors red and blue. So if you want to know which is the vena cava and the aorta, or the renal artery and renal vein, look at the direction of the arrow. So if the arrow is going towards the kidney, it's the renal artery because it's transporting oxygenated blood. And if it's coming away from the kidney, like over here, it is the renal vein or the vena cava, because it is taking blood away from the kidneys. And as you know, veins carry blood towards the heart and away from the organ, and arteries carry blood away from the heart towards the organ. It's just simple basics. This is taken from Save My Exams. I really love their content. I subscribe to them or like signed up, registered, whatever you call it, for my full ninth and 10th, and I found it so useful, especially in the ninth when I needed to get my concepts clear. So I definitely recommend using Save My Exams. Anyways, so basically, you need to know the structure and function. So basically, a kidney is a bean-shaped organ that filters blood, which we will see how later. Ureters are tubes that connect the kidney to the bladder. Bladder is an organ that stores urine, as I said earlier. Urine is a mixture of excess water, extra water, salts, and urea, okay, as it is produced by the kidney. And urea, as mentioned below, is formed from amino acids in the liver. And the urethra is like the tube at the end that connects the bladder to the exterior where urine is released. That pretty much summarizes what we saw on the diagram in words. But a picture speaks 1,000 words, so I don't think you really need this, but yeah. Okay, now this is an important concept that I took a little time to understand, though it's honestly easy, so I don't know why I took that time, but I'll try and break it down for you. This is also from Save My Exams, okay? So changes in urine. So basically, the color and quantity of urine produced in the body changes according to the conditions that are posed by the environment or that you are, the circumstances you are under. Okay, so if you have, so if when you usually when you excrete a large amount of urine, you'd probably expect to excrete that amount because you probably know that you've been drinking a lot of water that day. So basically, the more urine you excrete means more dilute the urine is, which means there is a greater proportion of water is to urea, which means the color is lighter. It's a pale yellow, not a dark yellow, and the quantity is more. Okay, so it's pale yellow because urea is less concentrated due to a greater proportion of water present. When you excrete a small amount of urine, it's going to be darker because you have a greater concentration of urea as compared to water because of probably different circumstances. Okay, so just remember small quantities means that it's going to be more concentrated and darker because of less water. Okay, so now these are the reasons of the concentration of urea, of why the concentration of urine changes. Water intake, as I already explained before, supposing I have a bottle of like 1 liter bottle of water and I drink all of it like really fast, and then I excrete urine. The quantity is going to be a lot because I've just taken in a lot of water, so the body needs to get rid of excess water. As mentioned earlier, excess water, salts, urea is excreted. That's what forms urine. So there's a lot of excess water because the body doesn't need so much. So there'll be a greater volume of water as compared to urea and the mixture of urine. So it's going to be a dilute solution. Okay. So it's going to be pale yellow. Then your next factor is temperature. So basically, if it's a really hot day in summer, you're more likely to sweat because there is a process called homeostasis, by which the body regulates your temperature to maintain a constant internal environment. And so to maintain the temperature, one mechanism is to sweat. That is to lose water in the form of water vapor, because when the water evaporates, it cools down your body. As you know, evaporation has a cooling effect. So basically, when it's a really hot day and you're sweating a lot, you're losing more water by your skin by sweating than actually excreting it through urine. So so less water flows into the urine. Yes, less water makes up the urine. So therefore, the urine becomes more concentrated and darker yellow. Okay, that's pretty much it. And the last is exercise, which is pretty much the same. More exercise, more water is lost in sweat, and so smaller quantity of urine is produced because less water is available in the urine. It's a concentrated solution, which makes it dark yellow. Okay. This I find really like easy and broken down. So I hope this helped you because it really helped me as well. And I hope I was able to explain the concept to you. Okay, now the liver. This is an interesting topic actually. So as we learned, this definition I've placed here just from lifted it from your chapter 7. So assimilation is the movement of digested food molecules into the cells of the body where they are used, that becoming part of cells. So basically, as you know, you digest proteins to amino acids, correct? And your body absorbs the amino acids by the small intestine in a process called absorption. And then when the amino acids move to the cells of the body, they need to be used. Now they can't be used in the form of amino acids. The body needs proteins. So the amino acids again have to be converted to form proteins. Now how does this happen? the liver assimilates these amino acids into proteins. Okay, assimilation, it builds up the amino acids into proteins. Okay, so that is one thing that you again need to be able to state. The liver assimilates amino acids into proteins, and that's how they can be used by the body. Now, the liver also carries another process called deamination, which is extremely important. Definition comes with 3 marks. You state the term, you get 1 mark. That's a separate question. Stating the term is a separate question. Definition, 3 marks. So basically, deamination is the removal of the nitrogen-containing group of amino acids to produce glycogen and urea. So as you know, every acid has a formula. So amino acids have nitrogen, hydrogen, carbon, and oxygen. Okay. So it basically, deamination extracts this nitrogen, whatever nitrogen atoms are there, the group of nitrogen from the amino acids, and this produces 2 products, glycogen and urea. Your syllabus does not require you to know glycogen, but I always added it to my answer because it just made it feel more complete. So maybe if you want to give importance to like certain words, you can probably write to produce urea and glycogen, because urea is a crucial part of this answer. Deamination produces urea from amino acids, removing the nitrogen containing group. Okay, so I suggest you learn this definition well, all the bold terms, etc. Very important, trust me. So that was basically your liver. Now we're gonna go on to your kidney, the structure of the kidney. I always found this really easy to draw. Like I used to draw this for my friends in school when we had to solve past papers together, and I used to feel like such an artist. Okay, but still, anyways, yeah, no one does that anymore. I know, you can comment how cringe I am. Okay, so basically, this is your kidney. Okay, this outer portion is your cortex. Okay, the inner portion is your medulla. Okay, here's your pelvis. Renal artery, renal vein. You'll get arrows here to identify, like going in is renal artery, coming out is renal vein. So it's probably gonna be like this. I'll just like draw it out for you. So renal artery, there'll be an arrow like this, and renal vein, there'll be an arrow like this. Okay. Then you have your ureter, which branches out from here. You don't have to know what pyramids are. This is okay. That one's okay, fine. That's basically it. Cortex, medulla, pelvis, renal artery, renal vein, and ureter. You have to know these, okay? I'll highlight them for you. So I guess it will be easier. So renal vein, renal artery, pelvis, ureter. I find it very important. I find highlighting helpful when you're doing it correctly, okay? That is basically your system. Now, when you have to draw it, just remember it's a bean shape. So I usually drew it like this, like much better, because like on paper and pencil it's different. And then I used to draw it like this. Okay, somewhat like that. And then the ureter usually has these bumps here. And usually you're supposed to label the cortex and the medulla and the ureter. They've come in, I think, the 2019 41 May June paper. You can check that out. If you want to practice excretion, it's a brilliant paper to practice excretion for all, because it has a whole question with a drawing. So yeah. Okay, now you need to be able to outline the structure and functioning of a kidney tubule. Now this is like lifted from your syllabus, but I found it important to like use the exact same wording. Okay, so basically the role of glomerulus in the filtration from the blood or water. So basically in the kidney, in the cortex, you have these folded structures called the glomeruli. Okay, so these have pores in them. Okay, and that is where ultrafiltration occurs. Ultrafiltration is basically when your solutions, like this solution, is filtered through these pores. Okay, so yeah, so blood is filtered through the glomeruli, and water, glucose, urea, and salts passes through. Now here it's important to understand that proteins do not pass through, because proteins are too big. They're molecules that are huge for the pores, and so they don't have the ability to pass through these pores in the glomeruli. Okay, but water, glucose, urea, and salts are filtered through it. They form the filtrate of the solution, and the proteins form the residue with the blood. Okay. Then you have the tubule and the absorption. Okay, so then there is a renal capsule and a renal tubule. So from the glomeruli, blood at a high pressure is forced out of the glomeruli, and proteins stays back in it, in the blood. Okay. The solution of water, glucose, urea, and salts passes through the renal capsule and tubule, where selective reabsorption happens. Selective means only certain substances are absorbed back into the blood. So these include glucose, okay, most of the water and some salts, okay, important. Most of the glucose is your first one, glucose always, because glucose gives you energy, okay, and this causes the concentration of, leading to the concentration of urea, okay, yeah, and this causes the concentration of urea to build up in urine because other substances are being extracted, okay. Um, so basically, this is what happened. Okay, you don't have to know the details, but in the renal capsule and the renal tubule, glucose, water, and salts are reabsorbed back into the blood. Now, at this point here, active transport occurs where molecules travel from a lower concentration to a higher concentration against the concentration gradient using energy from respiration. Very important. I got this wrong in one of my MCQs and one of my exams in grade 10, and I really felt bad. Okay. So just remember it's active transport in the excretory system, okay, selective reabsorption in the renal capsule and renal tubule, and ultrafiltration in the glomeruli, okay. So it's 4-star from the glomeruli to the renal capsule, and then as it travels to the tubule, selective reabsorption takes place. That's pretty much it, your major processes. Okay, so dialysis extremely important in IGCSE. You get it for like 5 to 6 marks just to state the process. Okay, so basically, blood is taken from your artery and returned to your vein. Okay, in dialysis. Okay, as you can see. So basically, dialysis it has a, so basically in your dialysis you have a fluid which contains the same which contains certain substances. Okay, let's say that. So first it contains glucose. Glucose is at the same concentration as the blood. Okay, so you have glucose in the dialysis machine, which is at the same concentration of glucose as the blood. Okay, which is same. I'm going to write same as equal to. Okay, then you have salts. Okay, which is again at the same concentration in the blood. And last, you have so basically there are 3 substances it can contain, which is glucose, salts, and urea, but it never contains urea. So basically, and let's just put it that way. So how does it work? So blood is drawn out from your artery through a tube. Okay, it is pumped, okay, into your dialysis tubing. Here, there is a partially permeable membrane in the dialysis machine, and dialysis fluid moves in to this membrane, okay, moves into this structure. Okay, now because the glucose concentration is the same inside the blood and outside the blood in the dialysis machine, there is no diffusion or movement of glucose molecules into the blood or out of the blood. Okay, there's no concentration gradient at all. It's the same concentration inside and out. So glucose doesn't move at all. So the patient does not lose glucose or gain glucose. It's the same as the normal body concentration. Same with salts. No concentration gradient, doesn't lose salts or gain salts. It's perfect. Now, why doesn't dialysis ever have urea? That is because there's no urea from the blood diffuses out from a higher concentration to a lower concentration gradient, okay, down the concentration gradient across this membrane, okay, into the fluid, the dialysis fluid. And what happens is that the blood in the body, the blood from the body is free of urea and it is purified. Toxic substances are removed. Usually dialysis occurs when a patient has kidney failure or something and it's a buildup of toxic waste substances. So these processes cannot happen, but here this dialysis allows it to happen. It may also have an anticoagulant, which basically prevents blood clotting, and there's a blood pump to keep the blood moving. Okay, the dialysis fluid is replaced and refreshed every 3 to 4 hours to ensure that the concentration gradients are maintained. Okay, so this is basically your dialysis. So for stating the concentration of glucose, salts, and urea each, you get like 3 marks. For stating the diffusion part, which diffuses, which does not, you get another 2 to 3 marks. Okay, and you need to like it's drawn out from the artery, return to the vein, all of that. You need to be able to state. Okay, that was basically your dialysis. And now the last part of your chapter is advantages and disadvantages, or like dialysis versus transplant. Okay, very important. So yeah, this picture was taken from Google. It had the most similar answers to what your mark scheme requires. So we're just gonna go through that. Okay. So kidney transplant is basically replacing your kidney using someone else's kidneys, a donor. You may have a kidney donor. So for example, I love Selena Gomez, so I'm just going to use her example. She had lupin, so she needed another kidney because of excess alcohol. So she needed another kidney. So her friend, so you can survive on one kidney. Okay, it is possible. Okay, so her friend decided to extract her own kidney and like donate it to Selena Gomez. Okay, and Selena Gomez now has her friend's kidney in her. That's really sweet. What a move. Commendable. Anyways. So kidney transplant, you can eat what you like. You lead a normal lifestyle. You go back to your normal lifestyle and you're free from restriction. You don't have to keep visiting the doctor anymore. Okay. Dialysis can be expensive. You have all those disadvantages. Dialysis can be expensive. Dialysis machine is free for others to use. But your disadvantages are that there is a lack of donors. Okay, you have to sometimes use, so basically the body can reject a kidney if it's not like, if it's not something that the, like, it depends on your blood group and the type of cells produced and everything, and it's foreign, right? So the body is likely to reject it. So the use of immunosuppressant drugs, so will have to be, so immunosuppressant drugs will have to be used, and that could that's not a very good option at all. That's one. And then you have the risk of rejection of the kidney, which basically leads to this. Okay. And then again, sometimes transplants are not accepted by social customs. Okay, so they may be expensive as well. So those are disadvantages. Now for dialysis, it's ready available. There's no risk of rejection. You don't need medicine. But yeah, your disadvantage is that it takes a long time. Regular treatments are required. Toxins build up between treatments, and there are diet restrictions. So these are your basically advantages and disadvantages of dialysis versus transplant. You should know at least three in each. For the 6-mark question, you're ready. For the 3-mark question, you're ready. So yeah, that was basically your chapter. I hope you found this helpful. Not like, very honestly, not gonna lie. Excretion is my favorite chapter. It's the one chapter I dreaded the most, even though it's like 4 pages in my textbook. But it's a really important chapter. Comes for many marks, not only in your theory but also in your ATP. ATP you're expected to draw. Then you'll be like asked to compare the values of urea versus salts and proteins, write a whole analysis. You have 6-mark questions about them in your structured. Then in your MCQs you have like tables and charts about what is excreted where, what process is used like active transport, etc. So you need to know this chapter very clearly in and out, through and through. If you have any questions, please email me. I'll drop down my email below. Please like, share, and subscribe. Please like, share, and comment on this video, and subscribe to my YouTube channel. I hope this helped you. Let's get through this together.